Okay, if there are no questions, let me just start. All right. So, I mean, that's what neutral theory was. Okay, and there is as I shared in that email, uh, there has been a lot of debate in the last few decades. Really, I mean, I think. um what happened was that so 68 is the proposal of neutral theory, and Timoko Ota is the other person who is heavily involved in the proposal of the theory, and she was a postdoc with Kimura. And these theoretical developments take place, and 83 Kimura writes this book, Neutral Theory of Evolution. It's a technical book. I don't consider it a book which is very easy to read, but if you guys have done a course in population genetics, it becomes much more accessible. Anyway, so I actually don't know of anyone who offers a popgen course uh, on campus, but anyway. um, but then over the years, as more data became, as more molecular data became available and people tried to fit the premise of neutral theory with this data, it was apparent that there is some problem. And the next best proposal that came was what is called the nearly neutral theory of molecular evolution. In the years subsequent to publication of his book, Kimura was sort of adamant that uh, it is going to be that neutral theory is a correct representation of how evolution proceeds. But I think people sort of more aligned themselves with this newly proposed nearly neutral theory of molecular evolution, which said that there is a vast majority of mutations which are slightly deleterious. So the essays associated with them are less than zero, but are very small in magnitude. And if you consider a vast majority of mutational change as nearly neutral, so selection is not acting against them in a very forceful way. That is a useful way to explain uh, explain the explain the sort of the polymorphism and the allelic diversity that you observe in a population. Okay. Um, there is in the next section in course, which hopefully we'll begin today. We will read a paper by Roy Kishney and Daniel Hartle. This was a paper in 2007 in Science, which I think brings about the problems that are associated with coming up with a correct understanding of what type of uh, what is the diversity between mutation between beneficial deleterious and mutational beneficial deleterious and neutral mutations as they occur. All right. So I did want to, before we move to experimental evolution, I did want to cover these two aspects, two ways of looking at neutral mutation. The first one, uh, so in the recent past, there has been some reports where a neutral mutation has facilitated adaptation. Okay? And this is weird. I mean, if a neutral mutation facilitates adaptation, can you really call it a neutral mutation? Okay, and the way why this happens is because of epistasis. Okay, so in order to understand this, there are two useful, very useful geometric ways of there are two very useful geometric ways of looking at it, and we won't go into the underlying math of it. uh, but the geometry itself gives us a very intuitive picture of how this could happen. In the first one, this comes from a book that was published in 2011 by Andreas Wagner, who has done a lot of work in neutral variation and its role in adaptation. So Wagner says, so the way we should visualize this geometry is the following. Consider a fitness landscape. Okay, But we are not looking at the entire fitness landscape. We are just looking at a very, very small part of a fitness landscape. In this really small part of fitness landscape, there are two consecutive fitness levels. One is Fi, and the other one is Fi plus 1, which is greater than Fi. At the level Fi, there are a few sequences whose corresponding fitness is Fi, and at level Fi plus 1, again there are a few sequences whose level fitness is Fi plus 1. The population currently is at fitness level A, and from A, there is no sequence at the level fitness Fi plus 1, which is at a Hamming distance equal to 1 away from sequence A, right? So everything is at least Hamming distance 2 away from this sequence. What that essentially means that from here, you can't jump to fitness level Fi plus 1. But should any individual in this population acquire a neutral mutation and move from the sequence represented by node A to sequence represented by node B, now there is a beneficial mutation available which allows this individual to move to a higher fitness, and then selection can act on it. But unless this neutral mutation takes place, you cannot access Fi plus 1, right? So this is one way in which Wagner proposes that neutral mutations can actually facilitate adaptation. And this is and what you must recognize is that this is epistasis, because this mutation, so we can imagine that this particular mutation is at position number 100. So at position number 100 on the genome, A changes to T and you come here, right? And this mutation is at a position number 1000, and at position 1000, suppose G changes to C and you go to this, go to a higher fitness level. But if you introduce this G changing to C at position 1000 in this genome, you don't go to Fi plus 1, you go somewhere else, right? But if the same change occurs in this genotype, where at position A you don't have a A but you have a T, then the same change at position number 1000 brings about this change, and that's the definition of epistasis. So essentially what Wagner is saying is that neutral mutation can work via epistasis in allowing access to fitness levels which were previously inaccessible to the population when it was at node A. Does that make sense?
Yes.
All right. The other is maybe a little more technical, but I think a little more realistic representation. And this is a paper that was published by Jeremy Draghi and Josh Plotkin in Nature or Science, I think Nature, in 2010. And what they are arguing is the following. So let's just before we get to them, let's just build some intuition over it. They say that if you have a sequence of length L, you will have a corresponding sequence. You will have a corresponding fitness. And we saw that if you have a sequence of length L, then there are 4 to the power L sequences possible, right? And each of these 4 to the power L sequences is going to have a corresponding fitness, right? But many of the sequences are going to have the same fitness. I mean, when we were looking at fitness landscapes and their structures, we saw that a vast majority of the sequence space is actually just going to be 0. So fitness everywhere is going to be 0. So the corresponding, so 4 to the power L unique sequences will have some fitnesses. And obviously the number of fitnesses is going to be much smaller than 4 to the power L, because many sequences are going to have same mutation, either because their fitness is actually just 0, or because there is a neutral change between these fitnesses. So you have 4 to the power L sequences and some corresponding number of fitnesses, and there is a 1:1 association between these two. Well, many fitness levels can have the same, many sequences can have the same fitness also. So while there is this relationship between these two, now what's going to happen is, suppose you are at, so let me now arrange all these fitness levels that I have in a increasing order. So this is the lowest possible fitness of a genome of length L, and this is the highest possible fitness. This is F max, this is F min. F min is going to be 0 because you're just going to have a chemical if you have all A's and stuff like that. Many of these sequences are going to be 0. So, and in between you will have these fitness levels. So let me just arrange all of them in an ascending order. So I go from F min, which is 0, to F max. Now consider, so this is just a different representation of a fitness landscape. Now consider that you are at some fitness node Fi, right? So that Fi, and let's imagine that this is a non-zero fitness because we have a living population. So our starting population is Fi, which is somewhere in this broad band of F min to F max. Fi is here. Now, what can you say? So obviously, when this population, which is at fitness Fi, acquires one mutation, what are the possibilities regarding the new fitness levels of this mutant that has arisen? What can you say about them? Is the question clear? Yes, no, somebody?
Yes.
Can you repeat it, sir?
Yeah. So if we have a sequence of length L, we saw that there are 4 to the power L sequences possible, right? And these 4 to the power L sequences, each is going to have a corresponding fitness, right? Now there will be a minimum fitness which is going to be 0 because many of these sequences are not going to correspond to a living organism, so the minimum will be 0. But some sequence will also have a maximum fitness, and that's F max. So you have a range between 0 and F max. Does that make sense, Prachita?
Yes, sir.
Right. If that's clear, and then between this 0 and F max, you have a lot of fitness levels available, right? Some sequence corresponds to this fitness, some sequence corresponds to this, and so on and so forth, right? So you have all these. So consider this as a ladder going from fitness 0 to F max. Now let's consider a isogenic population which you are starting with, whose initial fitness is represented by one of these levels, which is called Fi. Does that make sense? Right, I take that as a yes. If you have an isogenic population whose fitness is Fi, now an individual of this population can acquire a point mutation, right? Can acquire a point mutation which will lead to a mutant arising. What can you say about the fitness of this mutant that has arisen in this population? Anybody? Or if that's not clear, let me know.
It can depend on the dn by ds, and either it can increase or decrease.
So yeah, the mutation can increase or decrease depending on whether the mutation that happened was a beneficial one or a deleterious one. What other possibility is there?
It can stay the same where it
It
is.
can stay the same if it was a neutral mutation, right? Great. So if that is the case, then what is your claim? That is the Are the chances of mutation going up more or down more? Suppose you have an individual at fitness Fi, and you were to randomly introduce a point mutation into this sequence. Uh, what is your expectation? Is the fitness likely to increase or decrease?
Since most mutations are deleterious, fitness would decrease, right?
There is a greater chance that fitness could decrease, right? And depending on, I mean, the proponents of neutral theory will say that there is a good chance that fitness might decrease. There's also a very good chance that fitness might stay the same, and there is a very small chance that fitness might actually increase also, right? People who think that actually beneficial mutations are not a negligible fraction but actually are a considerable fraction will say perhaps that there is an equal likelihood of fitness increasing, staying the same, or decreasing. Fine. Now Draghi and Plotkin do something clever here. What they say is that let's start from fitness Fi, and from fitness Fi, so let's say the total number of levels which are available is equal to n, which means f naught is one level, this is second level, third, fourth, 5, and so on and so forth. The total number of levels is, let's say, capital N, right? Now if you have an individual at Fi, I mean, again, use your intuition about this. If you have an individual at Fi, via acquisition of one mutation, can this individual access any of the n fitness levels which are available?
No,
Okay, what would your argument be?
it can only go to the nearest fitness level by one mutation.
Well,
Not okay, not exactly the nearest ones, but it
yeah,
can't go very far either.
yeah, sure, exactly. Not the nearest one, because the way this ladder is arranged, it doesn't necessarily mean that the neighboring steps on the ladder are at Hamming distance equal to 1 from each other, right?
Yes.
Yeah. So it can't go to all, right? It can't go to all. It can go to a subset of these n. And let us say that out of the n levels available, via just one mutation, uh, the mutant can go to p out of those n levels, right? And it could be anything, right? I mean, some of those p will be beneficial, some of them will be neutral, and some of them will be deleterious. Great. Does that make sense to everybody?
So it can't go like very far because of that it can go only till the Hamming distance 1, that's why.
Well, yes, it can only go to sequences which are at Hamming distance 1 from it. But the way this ladder is arranged doesn't necessarily mean that the way this ladder is arranged doesn't mean that if you are at Fi, sorry, if you're at Fi, this is at Hamming distance 1 and this is at Hamming distance 2. This is not the way this ladder is around. This is all possible fitness levels. So it's possible that, you know, this is at Hamming distance 1. I mean, you know, number of sequences is 4 to the power L, right? And we saw that if you are a particular sequence, the number of neighbors you have is 3L, right? So, and the number of levels you have is going to be n. So it's possible that there are actually many sequences which have the same possible fitness. So this is one fitness levels, and at this fitness level you can have many sequences available, right? So each of this is a fitness level. It doesn't have genotypic information. So this is actually many sequences which are at this level. So maybe one of the sequence at this level is at Hamming distance 1 from here. Maybe one of the sequences here is also at Hamming distance 1 from here. Maybe the many sequences here, none of them is at Hamming distance 1 because this is too far. Does that make sense?
Yes, sir.
Yeah. So, I mean, you can structure it accordingly, right? I'm just trying to give you a basic flavor of this. There are many details that need to be inserted into a representation such as this. before you actually start simulations. And now is the key step that they ask, and then I will ask you about plotting a certain graph. What they argue is, if you are at Fi, right, and this is the key bit, so just pay attention here. If you are at Fi, you can access this and this, and at the deleterious level, you can go to this and this, right? Suppose you acquire a neutral mutation here, right? So you are still at Fi, but your genome has changed now. You are a new genome. From this new genome, can you also access, is it necessary that you have to access the same beneficial and the same deleterious levels as the parent could? Does that question make sense?
So could you repeat the question?
Yeah, I'll repeat it. Anirudh, you had something?
No, are you just saying that after every mutation there is some kind of dynamic change between the sequences and the Fi levels in that same?
Let me just say that again, and let me know if that makes sense. So let's say Fi, let's say an individual is at Fi, and its Hamming distance 1 neighbors are this level, is a neutral mutation, and 2 which are deleterious in nature, right? So that's the spectrum of fitness levels where this individual can go via acquisition of one particular mutation. Now, what Draghi and Plotkin argue is that if this individual acquires a neutral mutation, right, that means this new progeny is at fitness level Fi, right? Because by definition this was a neutral mutation, so progeny is also at fitness level Fi. But now the mutations that this guy can acquire is going to facilitate movement to some beneficial and some deleterious levels. Are those beneficial and deleterious levels going to be the same beneficial and deleterious levels that were accessible to the parent, or there is bound to be some difference in the levels that this guy can access as compared to the parent? Shreya, does that make sense now?
Yes,
So
sir.
what is your answer?
Well, I don't think they'll be the
Why
same.
do you say that?
I don't know that it'll be a different set of mutations that are at a Hamming distance 1.
Correct. Exactly.
So
Yeah.
there's a chance that the fitness would be different.
Right. Sure. Anirudh, what were you saying? Say that again now.
No, I think I got what you were trying to say here.
Yeah. And what is your answer?
Yeah, the same thing, I think.
Yeah. Right. Because epistasis is going to, two factors are at play here. One, epistasis is going to kick in, and as we just saw from Wagner's representation, neutral mutation acquisition, because of epistasis, a neutral mutation can facilitate access to another fitness level that was previously inaccessible, right? And second is that one distance neighbors are going to be different, and you can think of it geometrically like that. So the sequences that you have access to are now very different from the sequences that you had access to from the parent sequence, and hence the set of sequences, the set of fitness levels that you can access from here is going to be different as compared to the set of fitness levels that you could access as a parent, right? That is all that Draghi and Plotkin did. And now, and now the final thing, so they run these simulations, right? And to run these simulations, you can assume stuff. You can say that the number of fitness levels is maybe 1000. The P is the number of fitness levels that each sequence can access, P is the number of fitness levels that you being at Fi can access in just a single mutation. So maybe if the number of fitness levels, total number of fitness levels is 1000, you can make p equal to 10, and so on and so forth, right? They need some measure of, so you simulate a population via Moran process and study its overall study its change in mean fitness with time. And the measure for them is how long does it take for the population which is starting at fi for one individual of this population to reach fmax. What is the time taken, either in terms of number of generations or real time, depending on how you frame your simulation. How long does it take for one member of this population to reach fmax according to the rules that we have just defined, right? Of course, we've left out many details, but those are details. We can structure them in many different ways. And the rate of adaptation can then be described as 1 upon time taken to reach the peak fitness. If there is a particular design of, if there is a particular structure which makes access to the peak fitness sooner, that obviously means that speed of adaptation is greater. So let me ask you this question now. Now we define, I'm sorry, there's many variables here. Now we define a variable called k. Basically, k is that if total number of fitness levels is 1000 and p is 50, and basically what I mean by p is that how many arrows go out of it. Okay, if p is 50, that means 50 arrows go out from it, and they can go at some fitness levels, right? And k is how, out of those 50, what fraction come back onto itself. Okay, so if k is equal to 1, that means all 50 via acquisition of a single mutation, all 50 come back to the same fitness level. There is no beneficial or deleterious mutations. All mutations are neutral at this particular fitness level. If k is equal to half, that means that half the mutations bring you back to the same fitness level, no change in fitness, and the other half are distributed, either being beneficial or deleterious. Does the meaning of k make sense?
Yeah.
Okay. If that is the case, then what do you think this graph looks like? So you can tune the variation. You can tune the variable k from 0 to 1. K equal to 0 means that there are no neutral mutations. Every time a mutation occurs, your fitness levels change. K equal to 1 means that all mutations are neutral, right? Every time a mutation occurs, your fitness level never changes. Y-axis is the speed of adaptation, which is just 1 upon time taken to reach peak fitness. If I run these simulations, right, what is this graph going to look like? And I want you to comment on two things. One, what is your critique of this? What do you think about this quantitative representation of evolution of a population? Are there glaring inconsistencies with which you view an evolutionary process of a population which are not captured by this model? Or are you fairly okay with how this was modeled? And second is, what do you think this graph is going to look like? Comments.
The graph will be negatively sloped.
Okay. So
Like more k is equal to 1 will have the least speed.
Why do you say that?
Because like if it is neutral, then it won't reach to the peak fitness.
Yeah.
Only if fi is the highest speed of fitness, then only it can be
Exactly.
that way.
Right. Exactly. So I mean, when k is equal to 1, essentially you're saying is your fitness can never change, right? So you are just stuck at fi forever, right? Perfect. But what about the other thing? That are you okay with the representation of this process?
I think it goes with the general
Yeah.
perception.
Yeah, I think it's a fairly accurate representation. Of course, you're never going to be able to capture all the intricacies of an evolutionary process in a framework which is as simple as this. But that's sort of the beauty of this, that within the simplicity of this, it does seem to be consistent with uh, at least how I view it. But if you do have discomfort with it, please share it. Okay, anybody else regarding any of these 2 points? What do you guys think about the graph?
How does this graph capture the negative or positive mutations? It only differentiates between, but the way I see it is that it only differentiates between neutral and non-neutral mutations.
Yeah. No, so that's the background part of the model that we haven't talked about. You are working with the assumption that so something like this. I mean, there are other rules that you need to define before you can actually run this simulation. So the other rule would be that of all the non-neutral mutations, this fraction is beneficial and this fraction is deleterious, right? So the beneficial and deleterious mutations, the slots, the levels of beneficial and deleterious mutations need to be assigned according to some rules, and those are the rules that we haven't talked about. But a simple way could be that we are going to say that of all the non-neutral mutations, 20% are beneficial, 80% are deleterious, or something to that effect. Does that make sense?
So this is going to remain constant after each mutation to reach the peak.
I'm sorry, say that again.
So if
Well, it is because it's a map, right? So what you are saying is that my starting fi sequence, right? I can go to fi plus 1 and I can go to fi plus 3. Similarly, I can go to these two deleterious mutation levels, right? If you were at fi plus 1, then I can go to fi plus 3, fi plus 4. So all that entire structure has to be defined a priori. Does that make sense?
mm,
I mean, think of it like a fitness landscape, right? The landscape, if the environment is constant, the landscape is a constant structure, right? The landscape is not changing at all. Only the population is migrating on this structure, but the landscape is fixed. It's the same here. The network is fixed, that if you are at fi plus 3, these are the levels you are going to access. Does that make sense?
yeah, so when you mean peak fitness over here, you mean
Fmax.
Fmax, right?
Yeah.
So in that case, as it moves, and you are assuming here that there are many mutations happening to reach Fmax, or are you?
Yeah. I'm going to just I'm just going to let the population evolve till somebody reaches a fix.
Right. So I think, like, the way I see it is there is also a good possibility that I mean, it doesn't necessarily have to be. What if one particular F val? No, I don't.
No. Go ahead.
No, it's
It
just
doesn't matter. It doesn't matter. Just go ahead.
like does it necessarily have to be that the next point is always in that ladder? Let's say in x-axis, right? Is it does it have to be that every point in the x-axis is always going to be decreasing in time taken to reach the
No, that's exactly the trajectory I'm asking you to predict. And if you do not think it will be decreasing, why?
because
But I want you to don't worry about how you're phrasing it. Just say it.
yeah, no, it doesn't feel like I mean, if I go to the next mutation, for example, in that case it's possible that they have a higher amount of beneficial mutations as compared to the deleterious mutations. Suppose if I go to the next F value. And at that point of time, it would seem that the rate to go to the next F would actually decrease or increase based on the F. At each value of F, I think that there's going to be a different beneficial set of mutations and different deleterious set of
Absolutely,
mutations.
yes.
So that would decide kind of like the slope or time taken to go to the next F value.
Yeah.
So that
I mean, there are other complexities also. Would you think that when you are low down in the ladder, you would have a certain fraction of mutations which are beneficial? Would you expect that when you're really near the top, do you have the same fraction of mutations which are beneficial now?
No, I mean, I think it's based on the decreasing.
Yeah, it's
diminishing.
based on where you are in the fitness levels, right? So you would expect that the fraction of mutations which are beneficial decreases as you go along up on the ladder. So there's that dynamic feature also, right? Okay. But I guess I'm not sure what you mean when you say. What was your initial?
Like there is an instantaneous flexibility at each point, I think.
Yeah, that's kind of what I'm going to say.
Yeah, and remember this, that acquisition of neutral mutations. Suppose I'm at Fi, right? And I'm a population, and I'm at Fi, and one of the individuals acquires a neutral mutation, right? So as a whole, before this mutation occurred, I had only access to two beneficial mutation levels. I had access to Fi plus 1 and Fi plus 3 when all of us were at Fi with the same sequence. But now one of the members of this population has acquired a neutral mutation. So this guy is also at Fi plus 1. I'm sorry, this guy is also at Fi. But because the genotypic sequence is different now, while the remaining n-1 among us still have access to only Fi plus 1 and Fi plus 3, this guy has access to other beneficial mutations, right? And depending on what access, so this guy has maybe Fi plus 2 is a great level to be at, because Fi plus 2 gets you straight to Fmax via acquisition of a single mutation. There is just some sort of epistatic relationship there. So because of this acquisition of a neutral mutation, this guy has access to Fi plus 2. So its route to reach to Fmax just became so much more easier than the rest of n-1 members of the population, right? So these sorts of complexities can come into being as well. Does that make sense at all?
Yeah, it does.
So tell me about the graph. What does the graph look like?
Honestly, I don't know.
That's
at higher Fmax, I believe that it should be lower, the values.
at higher Fmax.
Sorry, at higher Fs, closer to Fmax.
Yeah.
I would assume that the time taken would be slower to reach Fmax.
Yeah. No, it does reach slower, but
Yeah.
but we're not. But that's not the question, right? I mean, our question is something different. We have fixed Fi, and we let the population go at Fi, and Fi can be somewhere in the middle or maybe close to the bottom of the fitness landscape. And at Fi, we let the population go, and we just note how long it takes to reach Fmax, right? So everybody is going to slow down as we reach towards the peak of the thing. That's embedded in the rules in terms of access to new fitness levels, right? So, okay. Any other thoughts, Anirudh, or anybody else? Yeah, Akshat, go ahead.
Yeah, hump shape.
Huh? Hump shape.
Hump shape.
Yeah. You've read this paper, right?
Yeah.
Okay, sure. That doesn't
Not
count.
that I remember, but okay.
Oh, sure. Yeah. Sure. So, actually, I can't hear you, Akshat. It's not very clear.
Nah, nothing. Go ahead.
Okay, cool. So this is what it comes out to be. That speed of adaptation actually increases as the fraction of mutations which are neutral increase. And then after a certain point, it starts to decrease, right? So, I mean, Sangamitra, I think what you are suggesting is what would be the common perception, that as the fraction of neutral mutations increase, speed of adaptation should slow down. And I think that's why this was this is a purely modeling paper. That's why this paper made it to, I think it was Nature. Right? If it was a monotonically decreasing function, then it's not such an interesting result. But this is a very interesting result, that you get this non-monotonicity, that it's increasing and then decreasing. Right? And then why should this be so? Right? The reason for this, we can go back to Wagner's representation. Actually, this paper came before that. Is the following: that at low values of k, what is going to happen is, in Wagner's representation, if population is here, so remember we are discussing low k, very few mutations which are neutral in nature. If population is here, and in order to move a fitness level up, you need to be here and not here, right? But this neutral mutation is very rare because k is very low, and this is sort of the only accessible path from here to reach fmax, for instance. Then you have to wait a really long time before this neutral mutation is acquired by somebody, such that this fitness level becomes accessible. As a result of this, the time you take is actually really long before you are able to transition from this fitness level to this one. As a result, when you increase at low values of k, as you increase the value of k, the time taken to reach fmax actually decreases, and hence speed of adaptation increases, because you're limited by the fact that this mutation is not taking place in the population. At high levels of k, what is happening is that if your population is here, neutral mutation is essentially every mutation is neutral. So the population is just continuously roaming around. Members are just roaming around from one level to another, and it's actually you have to wait a really long time for this beneficial mutation to take place. And if you're here, you need this and this to take place to be able to reach fmax. And what is happening at low k is that you get stuck because this isn't happening, and at high k you get stuck because this is all that's happening, this isn't happening anymore. And the dynamics of this play out in such a fashion that there is a sweet spot in between, where if the k is very close to this critical value, then you have speed of adaptation which is the maximum permissible in this particular framework. Does that make sense?
Yes, sir.
Yeah, right. So again, I think, so that's another way where neutral mutations can help. But I think the sort of the beauty associated with this work is I mean, I don't think the work is extraordinarily complex or anything, but the beauty associated with this is that being able to think in simple terms when it comes to quantitative representations of evolutionary processes. And I think those of you who are either working or interested in working with computational systems in evolutionary biology, the challenge is to, because it's inordinately complex. I mean, we can't even begin to comprehend the complexity associated with these systems. But the challenge in quantitative representation of these systems is that can you come up with representations which, for the most part, intuitively agree with the intuition about these processes that you have. It doesn't have to be exact, because it will never be exact. And then can you draw some predictions associated with these systems? But the first part, the representation of just thinking in those terms of a ladder and thinking that by picking up a neutral mutation, the access to levels changes now. That to me is the real key bit of thinking, which seems easy to think, which seems easy to agree with once you know, but I think is really hard if you are developing the model from first principles. So I think that's the reason why I really like this paper and the way of thinking behind it. All right, last bit about synonymous mutations, because we were talking about dn by ds. And if you do get a time, go through some of the references, especially that in 1996, one of the references that I shared in after the last class is this paper by this guy called Michael Creekman. And if you to get a very good perspective of where neutral theory is today, and where dn by ds as a measure of neutrality is today, is this paper in 1996 by Michael Creekman. It's one of the references I shared, and it has a title that neutral theory is dead, long live the neutral theory. That's a very useful perspective of how you can think about neutral theory when you're applying it to systems. All right, very briefly, dn by ds assumes, an analysis of dn by ds assumes that any non-synonymous mutation is either beneficial or deleterious. So remember, the species divergence took place, and we are comparing sequence of a gene between these two species, and there are some non-synonymous and there are some synonymous changes. And the assumption is that the non-synonymous change is something to do with adaptation. It's an adaptive change, whereas the synonymous change is a neutral change. And there has been a lot of evidence in the last 10 years that synonymous mutations actually are not neutral. Okay? And these are just, I'm just listing some of the mechanisms that people have discovered where synonymous mutations are actually not neutral at all, and they have adaptive value. And there have been some experimental evolution studies also. Let me come back to that. So I've just listed some ways by which synonymous mutations can have a fitness effect. The first is a synonymous mutation can play with mRNA stability. So if you have an mRNA is obviously extremely unstable, extremely reactive, right? So their half-lives are not very long. If you have a synonymous mutation, you are still coding for the same amino acid chain, but the structure of the mRNA can change because of this synonymous mutation. And if that change in structure brings about a change in the half-life associated with the mRNA, then obviously you're not going to get the same amounts of protein. And while you are encoding for the same protein, the amounts of protein are going to be different, and hence you're just not, your fitness levels are going to change despite the mutation being a synonymous change. Right? That's pretty straightforward. The second one is that if you have a codon change, you to incorporate any amino acid, for most amino acids, you have a choice that which codon that you want to use to incorporate that amino acid. And what would happen is that serine, for instance, has six codons to choose from whenever you want to incorporate a serine amino acid in a chain. And every organism has a bias in terms of that it likes to use one or two of the codons more than the others. And these biases can be really strong. Serine in E. coli, for instance, is so biased that one of the codons is used close to 40% of the times, and one of the other codons is used 5 or 6% of the times. So there is a factor of almost 8 between which codon to use whenever you want to incorporate serine. And the same is true for other amino acids. I mean, there are some where the differences aren't very huge, Phenylalanine, for instance, has two codons, and they're roughly used 60% of the times and 40% of the times. So the factor there is about 1.5. But in many amino acids, the differences are actually quite stark, up to a factor of 10 also. Now, how does this change things? This can change things in many ways. The general rule of thumb is that more frequently used codons are translated faster, right? So there is a stalling time. If you have a transcript and you have an mRNA, if you have a ribosome moving on this transcript, at every codon, there is a slight pause that's associated with the ribosome while it's waiting for a tRNA, which is charged with the corresponding amino acid, to come to bring this amino acid to this growing peptide change, transfer that amino acid from tRNA to the ribosome, which then gets incorporated in a growing peptide change, and then the ribosome moves to the next codon. The rule of thumb is that if you are making a protein which is using codons which are preferred by that organism, then translation proceeds at a faster rate. And if translation proceeds at a faster rate, you are going to get more of the protein as compared to if you were encoding the same amino acid chain with respect to if you are encoding the same amino acid chain using codons which are rarely used in the genome. By rarely used, I mean those 5% and 6%. They aren't rare by any means, but the general terminology is frequently used and rarely used codons. So again, by choice of codons, even though you're incorporating for the same amino acid chain, just because you altered the rate of translation, you are changing the amount of protein that's going to get resulted, and hence that's going to lead to a fitness change. The third one is you can actually alter the protein amount by changing access of the ribosome. So you can have synonymous mutations which change the structure of the mRNA such that the ribosome has trouble accessing the ribosome binding site and translation to get initiated. So mRNA transcripts which have a really tight 5' structure are usually not translated very strongly, because translation initiation is one of the limiting steps, and if that gets slowed down, then again the protein amounts suffer, right? Just on the previous point, the use of codons, and I said that proteins which use frequently used codons are translated faster. And it turns out that most of the proteins, if you sort of do a consensus study and you look at the codon usage in genes which are transcribed and translated to lead to a lot of protein compared to genes which do not result in a lot of protein. I mean, the cell doesn't need ribosome, for instance. Ribosomal proteins, you need a ton of them, whereas some other protein, you don't need a whole lot of it. Just a little bit is enough. So you will, and if you study the codon usage between these two groups of proteins, you will find that ribosomal proteins use frequently used codons, whereas these proteins which are not required in very high amounts use rarely used codons. So there is that correlation that if you want to make a lot of one protein, one of the strategies is you use codons which are frequently used such that they would get translated faster. And the last bit is actually something that's very recent and is actually a fascinating area developing, is that protein translation by changing this is sort of tricky. Let me just use this wire. Protein translation via use of different codons can actually lead to different structures of the protein. Okay? So the coding sequence is the same in terms of what the amino acid sequence being encoded is, yet the same amino acid sequence, depending on which codon you are using, can get translated into two different 3D structures of proteins. And the reason that happens is the following: that if you have a protein sequence, sorry, if you have an mRNA which is using codons which are translated very fast, then what's going to happen is the ribosome is going to move very fast, and this is going to result in this amino acid chain, and this amino acid chain is then going to fold into its structure, and this is going to do its catalytic job or whatever physiological function it serves inside the cell, right? Now imagine this mRNA transcript, it encodes for the same amino acids, but in the middle of it, I incorporate codons which are rarely used by the organism. Okay, so the original transcript was such that all the codons were highly used codons, but now I incorporate 5 codons here which are rarely used. And what is going to happen is, now as the ribosome moves, it's moving pretty fast, but the ribosome stalls here, and there's going to be a pause here because it's taking time to incorporate those amino acids. But in the meantime, this is the peptide chain that has already been translated. This is not going to hang around inside the cell waiting for the translation to be completed and then protein folding to take place. Because the ribosome is stuck, this has already started folding, and this folds into one structure. And by the time the ribosome comes out of this translational pause and completes the translation, you have this other peptide chain which has now been translated, and now this one is no longer available for folding because it's already been folding. Now the remaining thing gets folded into another structure, and hence by change of use of codons, you get 2 different protein structures with the same amino acid sequences. Okay, so this is another way in which synonymous mutations can actually impact structure and function of the protein. Does that make sense? Any questions with any of these aspects of synonymous mutations?
We never take this into account when we do bioinformatics.
Never.
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 Okay.
Yeah. So I mean, especially the last one, right? I mean, it has come up really in a really big way in the last few years, because now we know of examples which exhibit this property, and now one of the implications of that is really big in cancer, understanding cancer, because a synonymous mutation per se might not be doing something, but then if a synonymous mutation is present in conjunction with another non-synonymous mutation, that could lead to malfunctioning of the protein and cancer. So there is a lot of cancer data where there are synonymous mutations being found, and they had been always ignored up until maybe 3-4 years back. But now people are going back and examining all these synonymous mutations from all of these contexts, that we just talked about, especially the protein folding ones, because then, I mean, it's a different structure, and these alternatively folded proteins can just be doing different things inside the cell. Any other questions?
So, can we say there is, what about the selection pressure in this context?
In what context?
Synonymous mutations.
Yeah. So again,
It's kind of random, right?
What is random?
I mean, what does it depend on, like that?
So imagine this. I'm making up an example. Imagine this. Suppose you are growing a population of bacteria in the presence of a certain antibiotic. Okay. Now this bacteria is making an enzyme which is being made in a really small amounts such that this amount of enzyme that is being made is barely able to degrade the antibiotic that enters the cell, and hence the cell can survive. Is the context clear?
Yeah.
Now you evolve this population in this environment which contains this antibiotic. Tell me what would be an adaptive response of the population? What would be a good way for this population to evolve such that it counters the threat of the antibiotic?
What was the last part of it? Like you expose it to?
So the population is growing in an antibiotic, but this population does have an enzyme which degrades the antibiotic. The problem is that the enzyme is being made in such a small amount that it barely enables the enzyme, it barely results in enough enzyme to degrade the antibiotic. Right? The amounts of the enzyme which degrades the antibiotic are not sufficient basically. Okay. So if you were to evolve this population in such a context, what do you think would be a good adaptive response? Can you imagine a certain mutation which would help the population survive better? Or what does the cell need to do in your opinion to be able to counter the threat of the antibiotic?
Like the enzyme should be in that level or less.
Less or more?
Less. I mean,
Why?
enzyme should be more, yeah. So that the
Ah,
whole effect will be less, yeah.
right. So you need to increase the enzyme amounts.
Yeah.
Now tell me a mutation which would increase the enzyme amount. Either the enzyme amount needs to increase or the kcat associated with the enzyme needs to increase.
Either it should be a neutral mutation so that like nothing changes,
But
or
neutral mutation, nothing changes, so that doesn't help, no?
more of
No, don't think in terms of neutral mutation or non-neutral mutation. You just tell me. You tell me any amount. Yeah, Anjani, go ahead.
So it can optimize its codon so that more of the amount of protein can be synthesized.
Yeah, so essentially, yeah, I mean the mechanism comes later. Essentially the challenge is that it needs to make more of that enzyme. Now, what are some of the mutations that would enable this?
So
One of the
like you explained, the codons, some codons are more favorable than others.
yeah.
So if the mutations that organism is acquiring for the adaptation purpose, it can optimize those codons.
Yeah, right. So I mean a lot of things can happen. Essentially what needs to happen is the cell needs to do one of two things. One, it either needs to make more of the enzyme. Second, it can make the same amount of enzyme, but it can mutate the enzyme such that the kcat of the enzyme increases. That means it's able to degrade more molecules of the antibiotic per unit time. If it was degrading 10 molecules per second, it may be, if its catalytic property improves, it's degrading 20 molecules. Essentially, put together, you need to have enough enzyme capacity inside the cell such that more of the antibiotic is degraded. Now, this can be accomplished by change in a promoter sequence, right? The promoter mutation can take place such that transcription is more of transcription is happening, resulting in more protein. Or it can change in a mutation which changes the mRNA structure such that ribosome comes in and binds better. This mRNA structure can be changed via acquisition of synonymous or non-synonymous mutation. It could happen either way as long as the function of the coding sequence is not compromised, right? You could have changes in codons such that translation speed is enhanced, and so on and so forth, right? So I give this example because a few years back was the first experimental study where you performed evolution in lab and you actually analyzed what mutation, I mean, this is the section that we are entering into, performing experimental evolution in lab. But this was the so what do? Typically, not typically, every time, what you do is you start with an isogenic population. You grow cells in a particular environment which you are interested in because of whatever reason, and obviously the population is going to adapt in this environment. This was a study that came out maybe 5 years back in Nature Communications, that people evolved not equal pseudomonas in presence of this antibiotic, right? And at the end of the experiment, obviously because natural selection has been acting through these generations, you have a population which is better adapted to survive in this antibiotic environment than what you started with. And then what you do is you take your ancestral population, you take your evolved population, you sequence them both, and you try and compare what mutations happened which facilitated this physiological change of the population being able to better survive in the presence of this stress, which was antibiotic in this case. And what these guys found was that it was actually a synonymous mutation in the coding region of the gene, which allowed for enhanced amounts of protein molecules, which conferred a better resistance to the population, right? So that was the first example of you isolating a synonymous mutation where from a evolution experiment. Prior to that, the logic had been, prior to that, these claims about synonymous mutations had been made in the following context, where you take a particular gene, you change it yourself by incorporating synonymous mutation and studying whether there has been any change in the physiological performance of the cell. So these mutations did not arise by themselves, but you were studying them by putting in an alternate sequence of the gene that you were interested in. And since then, there have been more experiments which have isolated synonymous mutations as being the cause of an adaptive benefit that a population has acquired. So, Sangamitra, does that answer your question? I'm actually forgot your question.
Yeah, like I was asking, yeah, it does. Like I was asking whether, like, how does selection like happen in this context?
Yeah, so these are the contexts, right?
Yeah,
I mean, you know, so it can play out in any number of ways.
right.
The interesting thing is that suppose, you know, all of us do that pseudomonas experiment, evolve the same ancestral population of pseudomonas in the same environment, everything is identical. The complexity about an evolutionary experiment is going to be that there are 10, 11, 12 of us. It is possible that, you know, we do this year, and maybe 1 out of 12% amongst us is going to find that it was a synonymous mutation which facilitated that change. For the 11 others, it was non-synonymous or some other sorts of or promoter changes or whatever was happening. Maybe in someone's case, maybe there was a duplication event. So you have 2 copies of the gene which are both resulting in the protein that confers resistance, and that becomes adaptive because this duplication event facilitated more copies of the enzyme which help the cell deal with the antibiotic. And if I do this experiment with next year's class, maybe 5 people will see synonymous mutations arise. So adaptive paths is a complex problem, and the degree of repeatability in an evolutionary experiment at a genotypic level, that you are going to acquire the same mutations, there is very little consistency with regards to that. And again, this is something that we are going to enter into now when we discuss lessons from evolution experiments being performed in lab. But broadly, the consensus is that if your selection pressure is very specific in nature, like antibiotic resistance is a very specific resistance. You have a gene which confers resistance. You know that this is what it's needed to deal with this stress, and hence you're going to pick up you're going to pick up mutations in a very specific part of the chromosome which help you deal with it. Whereas if you have more general sorts of stresses, like, you know, if you're growing cells in low nitrogen or low carbon, I mean, these are processes that affects hundreds of genes. So the number of ways in which the cell can deal with it are also so many that predicting a priori that this is the evolutionary change that's going to take place uh, and help the cell deal with this particular environmental challenge, that becomes a really hard problem. So it also depends on the context in which you are performing this evolutionary experiment. Does that make sense?
Yes, sir.
How many of you have heard of GFP and RFP, or fluorescent proteins in general? Okay. All right. So since we are going to be moving into experimental evolution, and a lot of characterization of mutations is going to be done via use of these fluorescent proteins, let me just spend the remaining 10 minutes or so talking about this, and I'll tell you a really cool experiment which came out a few years back, demonstrating the effect of synonymous mutations. So because genetic code is universal, it means that if I have a gene which encodes for a certain amino acid chain, I can pick up that gene and put it in any organism I like. It would still encode for the same amino acid chain, right? Because the codon frequency might change. I might be using really favorable codons. The other organism doesn't like those codons or whatever. But in terms of information regarding the amino acid chain, remains the same. Right? A few decades ago, people found this gene in jellyfish, which makes the jellyfish glow. Okay. Now that for biotechnology applications is really big. These are called fluorescent proteins. Okay. And essentially what happens is that, so one of these is called a green fluorescent protein. Similarly, you have red, cherry, and all sorts of, then now people have engineered and made every conceivable color that you can think of. It started with green. Essentially what is happening is that jellyfish has this gene which obviously then gets transcribed and translated, and then you have a protein molecule, and this protein molecule has a certain, well, absorbs light of a certain wavelength and emits light at a certain wavelength. And because the emission is around the 520 530 nanometers, that invisible spectrum is in the green spectrum, and hence any cell which is expressing this gene, is making this protein, appears green in color, fluorescent green in color. And hence the gene which is responsible for this protein is called green fluorescent protein. Okay, so let me just show you a picture of this. This is GFP. You can look at this, but if you just look at images, these are mammalian cells. But if we go down, these are that this is a coconut tree. The leaves are green. That's E. coli expressing GFP, right? This is just jellyfish. These are rats expressing GFP. So all these are just biotechnology or science applications, studying biological systems using fluorescent proteins. Right. The point is, I can take this gene from jellyfish, put it in my system, and I will see my cells glow. Now, why would I need to put this GFP into E. coli, for instance? One of the ways to study this would be, suppose I find a gene of unknown function in E. coli. And E. coli has several genes for which we still do not know what is their function. Now, this gene obviously has a certain promoter which describes when it is going to be transcribed. And one of the ways of understanding the function of this unknown gene would be finding out what are the environmental conditions under which this gene is transcribed. So what I do via genetic manipulation is I take the promoter sequence of this gene with an unknown function, and I put a GFP under it, right? So now I have a genetic clone where the promoter is of this gene of unknown function, but it is not describing that gene of unknown function. It is sort of driving the expression of GFP. Now this genetic construct I will grow in 100 different environments, and the 100 different environments could be salt stress, oxygen stress, peroxide stress, low sugar, high sugar, low carbon, low nitrogen, anything, low phosphorus, anything. I'll grow these cells in 100 different environments. And now the cell doesn't know that it's GFP that is being driven underneath this promoter. So suppose this particular protein, this gene whose function I don't know yet, was involved in low phosphorus stress. Then in low phosphorus, maybe it helps the cell assimilate phosphate from the environment. Then in the cultures where phosphate was low, this promoter would be driving expression of GFP, right? As a result, the cells which were grown in low phosphate would make a lot of GFP and would be highly fluorescent, compared to the cells which were grown in abundance of phosphate. There, because there is phosphate, there is so much phosphate out there, the cell doesn't need to make that protein, and hence it won't need to express this gene. So I can compare this fluorescence level and immediately see that this gene seems to be made in environments where there is low phosphate. So perhaps the protein that I'm looking at is involved in somehow surviving and growing in low phosphate environments, and that becomes my starting point towards characterizing what this gene of unknown function could be doing inside the cell. Does that make sense to everybody? Those of you who had not heard of GFP?
Yes, sir.
Cool. So, and then, GFP is just one. Then people thought that, oh, wouldn't it be great if we could put two fluorescent proteins inside the cell and look at both happening? And this is done for a variety of functions. And a popular pair to use, and as a result of that, people have made lots of fluorescent. Now there is yellow, red, cherry, and many more. But green and red are a good choice, good pair to study, because the emission and excitation spectrum of these, they do not overlap with each other. Green and yellow is a bad choice. With your green and G and Y are right next to each other, so their emission and excitation overlap with each other. So you get a lot of noise from each other's signals. But green and red are a very good choice. So what in this paper they did, and now I'm coming back to the synonymous mutation bit. What they did was they created a transcript. So this is just a large transcript, okay? In this study, the first 3/4 of the gene is the 3/4 of a GFP protein, okay? So the sequence corresponds to GFP, but it's not the full GFP, it's 3/4 GFP. Then you have the remaining 1/4 GFP, right? And at the end of the transcript is 3/4 RFP, which is red fluorescent protein, which has been genetically engineered from GFP originally, right? So that is the construct that you have. That you have GFP 3/4, then the remaining 1/4 of GFP, and then 3/4 of RFP. Now, if this 1/4 protein folds with GFP, then you get green. But if this 1/4 folds with RFP, then you get red. So depending on what this 1/4 folds with, either you are going to see green cells or you are going to see red cells. Now these people wanted to study the effect of synonymous mutations. So what they do is, in one construct, they have this green 3/4 of the green fluorescent protein gene, and they put a lot of synonymous mutations here, such that translation will be slowed down here. Okay, so there will be a big pause here. And what happened in this case was that when this construct is translated, the ribosome is translating this really fast, right? But then it gets to this stage where you have put in these rare codons, so translation is slow. And because ribosome has now stalled here, the green fluorescent protein folds here, but this is incomplete, so this doesn't work. But then the ribosome comes out of it and then translates the rest of it very fast. But now this 1/4 now folds with RFP because this is the one that's available for folding. The green has already folded. So now what you see is actually cells which are fluorescing red and not green. In the other design, they change these synonymous codons to those codons which are actually translated very fast in E. coli, and moved the rare codons to after the 1/4 bit but before the red fluorescent protein 3/4 coding sequence. And now when this is going to get translated, the ribosome moves fast and stalls here. And now green is available to fold with the remaining 1/4 structure, and this folds. And now you have the red being translated later on, but it doesn't have the 1/4 available to fold with it, and hence you have incomplete folding of the red one. And as a result, when you now look at this thing, you actually see cells which are fluorescing green instead of red. So using fluorescent proteins, this was, I think, demonstrated in 2016 or 17. Using fluorescent proteins, this was a beautiful demonstration that by just altering the rates of translation, you can actually get sequence. You can actually get this coding sequence to fold in two completely different structures and serve two completely different physiological functions. Does that make sense?
Yes, sir.
All right, so we can stop for today. Wednesday we will start with experimental evolution, and we'll just talk about the basics of what would an experiment comprise, and how would we go about designing and how does the design need to represent what is the exact question that we are starting out to answer, is the reason why we are doing that evolution experiment, and so on and so forth. So we'll start with the preliminaries, and we'll just discuss the various ways. There are three primary ways of doing an experimental evolution design in lab. We'll discuss the three strategies, and then for the next 3 or 4 classes, we'll be discussing specific experiments and what are the lessons we learned from them. Okay. So if there are quick comments or questions, we can discuss them. Otherwise, let's stop for today. Okay, sure. So I will see you guys on Wednesday now. Bye for now.
Thank you, sir.
Bye.
Sir, I had a query.
Mm-hmm. Go ahead.
You said if we have two different codons, like synonymous codons,
Mm.
they translate into, like, they form two different kind of structures. They are two conformations.
They can form two different conformations. It's not.
Yeah.
It doesn't happen always. Yeah.
Okay. So if there's if second formation happens, is it transient? Like, does it go back to unfolding and fold into the proper conformation? Because if it is an enzyme, it has to go into the proper conformation to perform its function.
Good question. So, I mean, have you ever heard of the term energy landscape?
No, sir.
Okay. So, I mean, excellent question. So you can think of it like this: that, see, what does folding mean? Why is the, so tell me this? Why is the amino acid chain folding?
Like to it folds in a particular conformation because it's the least energy state, like the most comfortable.
Exactly. Absolutely. It's trying to minimize its free energy,
Yes, sir.
right? So now what is happening is that, suppose this gets translated really fast and it's going to fold into its energy minima configuration, right? So this is configuration 1. But suppose I incorporate rare codons here, so I get this particular configuration, that this folds into 1 and this folds into 1. So this is configuration 2, right? Now what is going to happen is that these can be represented as I think I have a figure for this. Let me just, yeah, there you go. Perfect. Oh, I'm sorry, you can't see. Let me share this screen. Can you see this?
Yes.
Yeah. So maybe the fully folded configuration was this configuration on the left, and where the two domains folded separately was this configuration on the right. Okay. Now, in order for the two configurations to switch from one to another, there is a big energy barrier here, right? So in this case, what is going to happen is that if you are a protein molecule which is stuck in one configuration, it's actually going to be pretty unlikely for you to switch into the other configuration, right? So as a result, you're sort of stuck in the configuration that in which you folded. But compare that with the two folded structures on the right. Now the energy barrier is actually very small, right? So there is going to be some sort of a dynamic equilibrium that's maintained between the two configurations in that particular case. So whether a folded configuration is sort of stuck in its state or whether it's a dynamic equilibrium between the two or multiple states is a question of what is the energy barrier between these two alternate states, right? And that is going to vary from case to case.
So if the energy barrier is high, there's still chance that chaperones are there which can help, which can act as a catalyst, like it can help in switching the one conformation to another. If the energy barrier is less, it can like itself go under the like equilibrium between the two states, right?
Yeah, except for the fact that, I mean, there are complexities here. Chaperones, the energy barrier could be high or low independent of whether chaperone is needed or not needed, right? So you could have proteins which fold by themselves and don't need chaperones but still have high energy barrier, so then you are stuck there. But even if you do have chaperones, it's not automatically given that chaperones are going to fold you into the correct state, right? The second case is that if you are a cytosolic protein, then if you're a cytosolic protein, then you could have this dynamic equilibrium between the two or more different states that you have. But if you're a membrane protein, if you are stuck in that wrong configuration, then there is no way out for you, right? So there is that dynamics and complexity also associated with this.
Thank you, sir.
Yeah. All right, bye, see you Wednesday.